Medicine Without Cruelty presents OMICS. Now, OMICS refers to a collection of disciplines in biology that study the collective characterization and quantification of biological molecules that translate into the structure, function, and dynamics of organisms. Now, OMICS are a collection of vast disciplines. You may spend an entire degree just learning about genomics and transcriptomics, for example, and a biochemist may spend their university career learning about proteomics and metabolomics. It is so important to have these cross disciplines work together. And with the power of AI and the context of non animal testing, omics technologies are highly impactful because they allow detailed insights into the biological responses without ever needing animals. So let's go through our first omics, and that's genomics, is the structure, function, evolution, and mapping of genomes. We talked about DNA in the previous section when we were talking about basic biology concepts. The entire DNA of an individual is known as a genome, and non-animal methods can use human cell cultures or tissues to study gene expression. For example, they could utilize genome editing, and CRISPR-Cas9 is a genome editing tool. You can think of these as molecular scissors, which can be used to cut the DNA at specific points. So we can edit our own DNA, and therefore it can be utilized in human cell cultures, as we know that the DNA, or the information of a cell, is located within the nucleus. Once we could get access to the DNA from there, we can use that to model diseases and test treatments. So, for example, say if you need to create a specific gene removal, you can use a CRISPR-Cas9 system to cut out your gene of interest. Transcriptomics is a study of transcripts, and that's our RNA. Now, DNA makes RNA, and RNA makes protein. Now, protein is the product of gene expression. If you remember from our previous section on DNA is a collection of recipes, and like a book, you could say, each recipe being a gene. So the product of those letters coming together is the protein, and the middleman is kind of the RNA. RNA allows scientists to measure any changes in gene expression when they're exposed to drugs or chemicals. And that's quite important because if we know whether a drug or chemical or any sort of toxin is having an effect on gene expression, we can know whether that change and what biological outcome will occur. And through high throughput RNA sequencing, or RNA-seq, we can assess the effects of these substances on human cells. Now, you imagine that with the power of AI to analyze the vast amount of RNA sequencing data that come from our genome, it means we can predict that these biological outcomes far quicker and far more accurately. Proteomics studies the complete set of proteins expressed within a biological system. So a proteome would be the entire protein that's expressed in a human body, and that's starting from the DNA. The DNA provides the recipe, and the eventual product is the protein. Human-based proteomics analyzes protein interactions, modifications, and functions without the need of animals. For instance, proteins from human-derived organoids are studied to understand drug mechanisms and toxic effects. And you may have heard of DeepMind AlphaFold. It's incredible. It predicts protein structures with remarkable accuracy, aiding drug discovery without ever needing animals. Metabolomics is another omics which focuses on the metabolites of small molecules which are involved in metabolic processes. Now, metabolomics uses human cell cultures or organoids to detect changes within the metabolic pathways due to the exposure of a substance. For example, say you might be studying metabolic changes within a liver organoid to predict drug-induced liver injury. Having that information allows you to understand what the changes occur due to the exposure of a substance. And it may mean that you're able to monitor potential side effects or produce drugs with fewer side effects. Genomics is an area which I've not previously touched on, but essentially our DNA has other modifications which are done to it, which means there's not a change in the actual structure of the DNA or the letters. There's no change to that, but there are modifications which can happen around it. Either it could potentially promote gene expression or it could stop gene expression. or silence them. And so these modifications can be histones, which are proteins that can surround the DNA, as you can see in the image there, or it could be, for example, other ways such as DNA methylation. But you could explore these modifications and histone modifications that don't change the DNA sequence but affect the gene expression. That will provide us quite detailed information about how drug-induced epigenetic changes can occur within human cells. And we can look at these patterns. Toxicogenomics combines the power of genomics, transcript proteomics, and metabolomics to study the effect of toxic substances on biological systems. Now, it's used extensively in human relevant toxicology studies because toxigenomics has the power of all these different disciplines to identify biomarkers or the hallmarks of toxicity in human cells and tissues. Now, one of the more exciting initiatives or applications of AI is the Tox21 initiative, which is a collaboration of multiple US agencies, and it integrates OMIC technologies and high throughput screening to assess the toxicity of chemicals without animal testing. By combining OMICS data with computational models, Tox21 predicts human responses to substances, advancing regulatory toxicology. Now, here are the following aspects of Tox21. It has a chemical library. The program has screened over 10,000 chemicals, including environmental pollutants, pharmaceuticals, pesticides, and industrial chemicals. Tox21 prioritizes chemicals for further testing based on their potential toxicity and relevance to human health. So they're classed into categories like high, medium, or low risk. Also, all the data generated by Tox21 is publicly available via platforms like the EPA's ToxCast dashboard. Researchers worldwide can access the data to develop new computational toxicology models. And the applications are far reaching. In regulatory toxicology, it can provide human relevant data to regulatory agencies like the EPA and FDA. It supports decision making for chemical safety assessments and approvals. In drug discovery and safety, it can help identify drug candidates with toxic properties early in the development process, and it enables safe and efficient drug development pipelines. For the environment, it can assess the impact of environmental chemicals on human health and guides policies for limiting human exposure to hazardous substances. In education and research, it serves as a rich resource for training in computational toxicology and alternative testing methods. It supports academic and industry research into innovative toxicology approaches. Now, the achievements have also been widespread. By successfully screening thousands of chemicals using the high throughput platforms, we can provide this rich source of data for education and further research. It also combines the HTS data with computational models to create predictive tools for toxicity. It's made all of this data freely available, and also it's demonstrated the feasibility of removing animal testing with human relevant methods. Now, if we think about the challenges and the future directions of it, there is a lack of human relevant data for certain chemical categories, and so we need to make sure that we expand the chemical library to include more complex substances. And also, we need to make sure that because there is a difficulty in replicating complex organ interactions and systemic effects, both in vitro, so in cell-based models, and in silico in computational models, we need to make sure that we improve multi-organ models, such as the multi-organ on a chip that we discussed in the previous section, and integrated testing systems. We can incorporate the advancements in AI, such as deep learning, to enhance predictive accuracy and collaborate globally to establish unified standards for alternative toxicology methods. We need to ensure regulatory bodies fully accept non-animal methods. Thank you for listening.